Happy 2025 and welcome to Life Science Success Podcast. My name is Don, and before we get started today, I just wanted to play a brief message from our sponsor.
This episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more. This episode.
Dr. Prakash Narayan is our guest today on the Life Science Success Podcast. He's a distinguished biotech executive with over 20 years of experience in leading IND and BLA-enabling programs from concept through clinical trials for heart, lung, kidney, and liver indications. So with that, welcome, Prakash. It's great to have you on.
Thank you, Don, and Happy New Year.
Somehow I moved on the screen here. So there we go. Now I'm actually not moved, but yeah, it's great to be here.
Thank you, Don. Happy New Year.
Yeah. So would you mind just telling everybody a little bit about how do you go from somebody who's pursuing a degree at Ohio State to a biotech executive?
Sure. First of all, hat tip to the Ohio State. We won the Rose Bowl last night, so it was a good moment. So how do I go from a biophysics degree to an executive? I think that's part of life, right? And that's part of the evolution or a journey. So my own journey started very traditionally. I took, I did my GRE and all that, came over to this country more than 34 years ago now. Graduated with a biophysics degree from the Ohio State University. And then I did the 2-year mandatory, if you will, postdoc at University of Madison, Wisconsin, and then University of Kentucky. I moved up to a research assistant professor non-tenure track at the University of Kentucky. But then I did not see myself progressing there, if you will. It's a very competitive field. It was even back then. You need those grants to come in and stuff like that. And also, I think money called. So I actually joined a biotech company on Long Island. This was 2002. And pretty much led programs there as first a principal scientist, if you will, and then moved up the ranks to become the VP of non-clinical R&D over there. I think some of the things that may have helped me back then is I think my written communication skills were and may still be very good. So that helped me. And I think in a company environment, they really look for a team player, somebody that fits in. They don't want you coming in and maybe disrupting them with your own ideas. They want you to deliver what they have in mind. And I think maybe I did that. So I kind of, between the communication skills, being a team player, I'm not tooting my own horn here, and maybe a people person, I think I rose through the ranks at Angion.
Yeah, I mean, it's really interesting to hear how people, sort of follow this non-traditional, non-linear path. But I think it's the way that all of us, kind of operate. That we, life somewhat happens, but in addition to that, you have, opportunities that you can clearly see as well. And it's good that your experience led you there. With your experience at Angion, how did it help you think about the ways that you grow a startup into a publicly traded company? How did that, also shape your perspective in the industry?
I think I want to reiterate what I think most people know. Graduate school does not prepare you for life, right? You pretty much have to take the plunge, and you learn along the way. You learn what to do, and you'll also learn, equally importantly, what not to do. Okay, and you have to be flexible. You have to adjust and stuff like that. So a culture shock, at least professionally for me, was coming out of graduate school or postdoc or academia into the biotech world. And I think what that taught me was A, to be nimble, to be flexible, keep your antenna up, keep your radar up, learn what's going on, learn what the market forces are. You may have a great idea, and but unless you can reduce it to practice, and unless that idea remains great, when it becomes reduced to practice, perhaps 15 years from when you thought of it, I think those are some of the things you need to learn very quickly. And I think it was a great experience at John. What we did was we went from literally a mom and pop shop. I think I was the first employee back then. And we were about four and a half, five people, half meaning one was a part-time employee. And we took it over about a 15-year period to a publicly traded company. Non-dilutive funding raises, a lot of due diligence visits, visits to the FDA. So you learn a lot of things along the way. And if nothing else, it prepares you, it makes you more resilient, it prepares you for the next chapter in your life, wherever that may be. So a great time at Angion.
Very good. Can you tell us about the process of taking a drug from discovery through IND studies to the FDA filings?
Yes. At Angion, at least, our bread and butter were small molecule therapeutics, and that's typically what, your Tylenol, your ibuprofen, they're all considered, your not Ozempic, but your Farxiga, etc., they're all considered small molecules. Okay. And that's pretty much the bread and butter of pharmaceutical industries today. It's the bulk. Uh, so that journey starts maybe with a concept, hey, I'm going to try to address this form of cancer because the need is either unmet or undermet. Okay. And then you start surveying the literature, you start seeing what's maybe already out there, what the weaknesses are. You do a SWOT analysis, if you will, right? So strengths, weaknesses, opportunities, and threats from a business perspective in trying to address the problem. You have a medicinal chemistry team, a computational biology team that essentially evaluates that target that is associated or maybe even causative for the disease. And what you do is you try to see if you can come up with something that can disrupt that target. How do you come up with something that can disrupt that target? Well, a couple different ways. You can try a fast follower approach. So if there's something already out there, but it's not, working great, or it has some side effects, you can come and try to maybe make adjustments to that molecule, small molecule that's already out there, while gaining intellectual property and improving upon its efficacy and reducing its side effects. So you have a big medicinal chemistry initiative, typically with a computational biologist who can do a lot of the in silico screening, kind of the first pass screening. So one is fast follower. The other is you can literally buy up small molecule library. So there are companies out there that sell, or make available to you for a fee, millions of small molecules. Some of them are as good as paint. They have no value, but something else may have value. And then what you do is with your medicinal chemistry team, you go from a hit to a lead. You start optimizing that small molecule. And by optimizing, I just don't mean making it more biologically efficient, but also making sure that at the end of the day, you capture intellectual property, because that's the currency in our business. Okay. Once that you have a hit, you also have to build a library of small molecules around it. So you have to build an intellectual property fence, so that nobody else can then come in and then say, hey, I made a small adjustment to your small molecule and they have, something better. So you build a library around that small molecule. And then you take it to in vitro, so cell-based assays, enzymatic assays. These are essentially test tube type or culture plate environments, and you see how your molecule performs. You give feedback to the medicinal chemistry team, hey, it was great, but it wasn't that potent. Can you make it a little bit more potent? By potent, I mean, can it work at a lower concentration? And then you make those adjustments, and once it passes muster with its that lead or a new lead, you then advance it to animal studies. So these are models of kidney disease, liver disease, heart disease in typically rodents, mice, and or rats. And you look at efficacy. In parallel, you start looking at other aspects called PK, PD, so pharmacokinetics, oral bioavailability if it's a chronic indication. Um, you start looking at plasma protein binding. You can take a drug, but if it pretty much doesn't circulate well, or it just binds to all the albumin in your blood, it's not going to have much activity. So you have a parallel track in addition to showing efficacy. PK/PD, you have a toxicology first run. I start escalating the dose in animals, seeing what happens to the animals. All this is done humanely with the proper regulations in place. And once that passes muster, you start getting into what are called IND enabling studies. So these are GXP, good laboratory practice, good chemical practice type studies. These can get very expensive, so you have to be darn sure that whatever you're bringing to that stage has checked off all those boxes. And once you have a profile where it's efficacious at this dose, but maybe has toxicity at a much, much higher dose, it's called a therapeutic index, where your efficacy occurs at a much lower dose than when it's toxic. You can maybe file an IND to do first in human trials. And that's a phase one study in healthy volunteers. If you pass muster there, you go to a phase two study, which is actually looking at efficacy and safety in a patient population. You have meetings with FDA. If that passes muster, your phase two study, then you go to a phase three, typically two registration phase three trials, and you take it from there. And if everything goes well, you file a IND, investigational new drug.
Very good. And when I looked at your past as well, you also have experience with cell-based therapies as well. How do they differ in terms of the work that you've done with small molecules, and then what unique challenges do cell-based therapies present?
Great question. I think it's literally night and day. I think with small molecules, it's more of a defined template. I think they're more predictable, if you will. You can figure out, if I take a milligram or a gram of a small molecule, you can figure out how it gets metabolized. It's literally chemistry. Okay. You can find out when that small molecule is getting cleared or eliminated. Is that clearance of that small molecule occurring through the kidneys, or is it occurring through the liver? So I think there are swim lanes already in place guiding a small molecule development, just simply because I'm going to say chemistry is easier than biology. With a cell-based asset, I think the challenge is a mechanism of action. What is that cell doing? Coming back to small molecule, it's very defined. You have a receptor that you either want to typically agonize, activate, or you have a pathway that you want to inhibit with a small molecule. With cell-based assets, you're not sure, it really doesn't bind to a receptor. You shove the cells in, and there's a lot of success with cell-based therapies. Don't get me wrong. In fact, the hematopoietic system, it's a lot of that, and the CAR Ts and all that. But with the cell-based, again, you don't know exactly what it's doing. You don't know how much of the cells you need. Again, with a small molecule, you can literally dose up milligrams per kilogram. With cell-based, do you put 1 million cells in? Do you put 10 million cells in? Yeah. Then clearance, what happens to the cell when it enters your body? So even before we get to that, you have to make sure the cell is allo friendly. It doesn't get rejected by your immune system. You want to know what happens when you deliver it into your body. Does it end up in the lot of the cells, including the mesenchymal stromal cells, they end up in the pulmonary space. Some of it gets cleared by the spleen. So those are variables that haven't really been chalked out. They're hard to figure out. Also with cell-based, you really can't tag it that easily with a label such that you can track it yeah, into the system. So I think the challenges are many, much easier, a path forward, if you will, with a small molecule. But that being said, cell-based therapies do have their space in medicine.
Yeah, absolutely. And I see a bright future as somebody who's not, somebody who spent a lot of time in the lab creating medicines. But yes, yeah, for sure. Your profile mentions also that you've used predictive analytics with empirical findings. Can you give an example of how this approach has informed your work?
Yeah, I think science has come a long way. I think last 30, 35 years, I think biomedical science has come, progressed tremendously. And most of our findings, or our not findings, I'm going to say most of the that yellow brick road that we use has already been paved for us by, I'm going to say, the oncology community. Uh, so what is happening now is that we have to show, when you show proof of concept, okay, you show that your small molecule or cell-based therapy works. What you try to do, and that's the empirical part, what you try to do is find the associated pathway, the pathway that is associated with the outcome. And what you do is try to figure out the mechanism of action of how your asset is working. Okay. Uh, and the idea behind that is not just to figure out how it works, which is very important. I think the agency, FDA, of course, wants to know what you are putting into a person. It's a very highly regulated industry. But the other aspect to figuring out mechanism of action is you can literally take your asset and repurpose it to another indication that is sharing that mechanism of action. Okay, so you're essentially now opening up the opportunity. You show proof of concept maybe in a gateway indication, and then find identify other areas where this pathway that your asset is modulating, whether it's agonizing or inhibiting. If there are other indications where those same pathways are playing a role, you can repurpose your cell-based therapy to that indication also. So I think the paradigm has shifted from treating a disease, if you will. The disease is essentially the end manifestation to actually treating a pathway, okay, that's causing the disease. So now we talk in terms of, for example, Humira, adalimumab, right? It's a TNF alpha inhibitor. It's an inflammatory pathway inhibitor. So if there are pathways, TNF alpha pathways that are playing roles, not just in rheumatoid arthritis, but something else, you can then repurpose a drug that's out there to those indications. So I think that has really caught on. It has captured the interest even of the agency, and I think that's where the future is today.
Very good. So whenever I look at your overall profile, it also your experience spans various disease areas like kidney, liver, and lung disease. How do you adapt your approach whenever you have to switch between these different focus areas?
Oh, that's a great question. Yeah, and in fact, let's just go back to the what I just spoke about. So let's say you have a drug, small molecule, cell-based, antibody, peptide, doesn't matter, and you find that you are using it to treat liver disease, but the pathway that it's engaging with is also a pathway that's playing a miscreant role, if you will, in a cohort of patients with lung disease. Okay, so now you want to take your drug and then repurpose it to that population of pulmonary patients that unfortunately have that pathway causing that disease. So the challenges are many. Okay, so the liver, typically what you do is for a liver type of indication, you want an oral pill. Okay, an orally bioavailable acid. For lung, that same asset should be inhalation route. Okay, so now you are trying to figure out how do you take a pill? You can't snort Tylenol, right? You can't snort ibuprofen. But for lung, what you want to do is you want to get that first pass exposure as much of your therapeutic agent into the lung, because believe me, whether it's a cell or a small molecule, the body starts dealing with it very quickly. It starts, okay, it doesn't recognize this. This is not a natural entity, and it starts trying to get rid of it. So for the lung, now you have to come up with the asset that engages the pathway of interest, but now you have to make it an inhalation type of pathway, inhalation route applicable via that route. Uh, Now, there are some other things that will may help you out. I think the bar is different as far as different indications are concerned. So, for example, with kidney, if your kidneys have failed, or you're in end-stage renal disease, you actually have an out, and that's called dialysis, right? It's not fun, but you can, 3 times a week, you can get dialyzed, and you can, live many years until hopefully you get a kidney transplant. There is no dialysis for the lung, okay? So, and there's no dialysis for the liver. So as far as, let's going back to the lung, the FDA will say, okay, it's a much, much more life threatening disease that you have, idiopathic pulmonary fibrosis. So the agency actually will maybe lower the bar for you in terms of if your asset is a little bit toxic. I think that bar will get lowered as far as something like cancer or idiopathic pulmonary fibrosis, those types of indications, because they are essentially you're a dead person walking. If you have that, your runway is very short. So you have to, I think, to answer your question, you have to not just say, hey, I found this in the, for the liver, let me just put it into the lung. There are tons of other variables playing a role, and you have to recognize all of that, have dialogue, bring in the pulmonary experts in this case, see what they have to say, see what the competition out there is in that space, and see if bridging a need there with your existing asset makes sense.
Very good. And can you explain the concept of precision medicine and how you've applied it to clinical trial inclusion criteria?
Yeah, so the precision medicine initiative, again, the playbook came from cancer. I think it came maybe even 25 years ago from the oncologist. And what they were able to show very nicely is that there are mutations, typically a mutation driving your disease within cancer. And if you address that, if you're able to maybe neutralize that mutation or address it in some way, you can have a better outcome. Excuse me. So what they did was they used strategic targeting, if you will, okay, to position a drug for, say, breast cancer, another drug for acute myeloid leukemia, something else for hepatocellular carcinoma that worked on the culprit mechanism that was causing the cancer. With other diseases, it's a little bit more challenging. Okay, there's no for the majority of the part, kidney disease is not driven by a mutation. Okay, what causes kidney disease is diabetes, hypertension, metabolic syndrome. So it's not a single mutation dependent disease. Although there are genetic causes of kidney disease, for polycystic kidney disease, for example. Liver disease, okay, there's something called MASLD, or previously known as NAFLD or NASH, and that's fatty liver disease, not of alcoholic origin. Again, that is a disorder largely due to a lifestyle of plenty, if you will. Okay, you take your calories in, it's a holiday period. Uh, you drink a little bit, and then your liver starts getting affected. So it's from a precision medicine perspective, it's not a single mutation causing these diseases, including idiopathic pulmonary fibrosis, but probably a constellation of factors. And I think the challenge there is to identify pathways that are dominant in these other diseases, in the liver, the kidney, the lung. And I think I gave an example. Let's say I have kidney disease, and we have figured out that it's being caused by TNF alpha. What might be doable is to then repurpose something that works against TNF alpha for my kidney disease. But if my kidney disease is largely driven by something called an interleukin, okay, IL-6 driven, then I want to be able to take something that neutralizes the IL-6 pathway. So that is what precision medicine essentially is. It came out of an initiative by Obama, I think 2009, the White House Precision Medicine Initiative. And I don't know if it's just if he said this or it's attributed to him, but essentially precision medicine is one size does not fit all. Okay, precision medicine is getting a drug to the right person at the right time every time. As an example of how we attempted to position or leverage precision medicine to treat kidney disease, we were looking at focal segmental glomerulosclerosis, which is a nephrotic syndrome type disorder, causes protein spilling into the urine, and it's a very aggressive form of disease that uh, causes your kidneys to fail eventually. We, under the aegis of the Department of Defense, we had a very large grant award, if you will, where we were able to identify, we had a small molecule in place, but we were able to identify which pathway that small molecule targeted, okay, in focal segmental glomerulosclerosis. And today there are biobanks, there are electronic health records. So we were able to match which patients share this pathway, okay, from existing biobanks. Okay, because the small molecule worked great in the animal studies. We were able to figure out from those animal studies which pathways that small molecule targets. And then we queried those patient biobanks, those tissue biobanks, those electronic health records, if you will, found out a population of patients, a cohort of patients that actually shared the same disease causing pathway that we saw in our animal studies. And we were able to say, hey, let's use our drug, not on everybody with focal segmental glomerulosclerosis, but that cohort that shares this mechanism where our drug acts on. So I think that was a great, it was very rewarding that we were able to apply what we had learned from oncology, and we were able to bring it to kidney disease.
Yeah, so important as well that that we can see those things through the animal studies and the data that's available. Um, One of the things that you've mentioned a few times is the your approach in terms of funding, and the fact that you've been successful in getting non-dilutive funding. I feel like a lot of the entrepreneurs that I talk to today, they get frustrated with kind of the long timelines to get non-dilutive funding, and so they just don't go get it. And to me, it's almost like the people that I've seen. I work with one researcher right now who's very successful. I feel like at getting non-dilutive funding, and it's carried the company through some really tough times whenever we haven't been able to find venture capital or others, other methods of funding, plus the fact that you're not having to give something up as a part of that. When you look at non-dilutive funding, how do you think about it, and then what advice would you give to biotech professionals? Because I'm certainly not the person to give that advice, but I also would look to somebody like you and just say there has to be considerations that people aren't making today that they should be.
I think you hit the nail on the head. There's the yin and the yang to non-dilutive funding. So the advantage, like you mentioned, is you don't have to give up a seat at the dinner table, right? Uh, the yang or the opposite end of that is it takes a long time to actually get those funds. But let's I think the way to look at it is to try to do both. Okay, the non-dilutive funding, I think what it does very nicely is it de-risks not de-risks, it distributes, right? I think that's the word I'm looking for. It distributes the risk of that investment across taxpayers of the United States. Okay. So the SBIR, NIH, SBR mechanism, if you will, and the National Science Foundation, and literally every TSA, they all have mechanisms to encourage entrepreneurs, and the funding for that can, it starts small, but it can actually blossom to many more dollars. And the idea is to maybe apply for non-dilutive funding, because it essentially distributes that risk. Each of us as taxpayers, we are paying very little towards somebody's idea. But what it can also do is bring your idea up to a point where it becomes of interest to potential investors. Okay, because investors, I think they look for exit strategies. They want to invest, but maybe they have 2 3 years timelines in their mind, and within that they want to make some money. So with non-dilutive funding, and I've written my share of SBR grants, I've gotten my share of SBR grants, and I've not gotten my share of SBR grants. I think the idea is to be able to do both. Go that route, see if you can, get non-dilutive funding, but also keep investors think about them in your back pocket, and maybe try to get a blend of both. So I think that would be my suggestion or advice.
Yeah, it seems like wise advice, especially given some of the challenges that a lot of companies face, and some companies I feel like would present themselves really well in an SBIR grant, but at the same time, I'm not the one who's necessarily leading that charge, and it just leads to a very interesting conversation overall.
Yeah,
Um, sure.
one more point. The SPR is not a charity. It's not a gift that keeps on giving. I think what they would like to do, and rightly so, is okay, here's some money, take your concept, bring it yay forward, and then actually apply for, private capital, see if the real world people out there are interested in what this asset, with the potential of this asset. So I think it's like a stepping stone, if you will, non-dilutive funding. But I think at some point you have to get off that and start, real world, Wall Street, or wherever else. Yeah.
Yeah, absolutely. And so there were two words in your bio that I feel like people may not understand. And before we hop into the next question, can you explain first of all what are transcriptomics, and then what are functionomics?
Great question. I apologize for using those buzzwords, if you will. So what has happened again from the last 30 years is that folks have gotten away from looking at a single, pathway or mechanism of action, hypothesis testing, if you will. They've gotten away from that to what is called a hypothesis. Let's just do a blast. Let's look at everything out there. Let's not bias ourselves, and then just see which pathways, or which aberrant pathways actually tip their hands. So back in the day, we look at a single receptor, see if it's involved, do a Western blot or an ELISA, and try to chase down if it's upregulated in disease. You want to downregulate that single pathway or receptor, and go that route. Transcriptomics is essentially a way to look at mRNA. So those are genes, if you will, that may be associated with the disease. And the nice thing about transcriptomics, or I think the word is omics, whether it's proteomics, metabolomics, transcriptomics, is to query literally everything out there. Okay. How do you know what? So you're looking at maybe 30, 40,000 variables right now instead of that one variable. So how do you know which of those 30, 40,000 variables, how many of them are playing a role in disease? That's where the functional part of the functionomics comes to play. You start seeing, a simple word, which correlates, which of those 30 or 40,000 genes, their expression levels correlate either inversely or directly with function of the organ. Okay, so if my kidney function is declining, and I'm beating up on TNF alpha today, but and my TNF alpha, pathway is going up, chances are there's an inverse relationship because, I won't say because that's causative, but there seems to be an association between TNF alpha going up and then my kidney function declining. Realize that I did not start by looking at TNF alpha. I'm looking at 30, 40,000 things. I'm looking at kidney function on this side, and I found that there's a very nice correlation, if you will, inverse between increasing TNF alpha levels and my declining renal function. So that's transcriptomics, looking at gene signatures, pathway signatures, and then the functionomics is actually correlating that to function, and then identifying, hey, okay, this looks like the culprit mechanism in this patient population in this disease. Let's try to attack it. Let's try to address it and see if it corrects the problem.
And do you think that is a part of, the way that people are looking at it is just because we have the tools today? Look, why wouldn't I look at everything that's available, given the tools that we have today and the ability to look at everything? Where before, with some of the other tools and mechanisms, you only had a very select choice that you had to make, and then you might be blinded to something that could actually get you later.
Absolutely. And I think you have segued into a favorite, topic of mine, which is we have heard of the Stone Age, we have heard of the Bronze Age, the Industrial Revolution. Don, I believe that over the last 30 years, I'd say mid 90s, when the internet started, becoming popular, till today, we are in some kind of an age. You can call it informatics age, digital, or whatever. Let's not worry about what we're going to call it. And it's exactly right. There are so many tools today. It's almost like technology is no longer the bottleneck. It's how you use it, what kind of creativity you bring to the table. Don't even get me started on AI and all that. We have so many options today that we didn't have back then, and this is essentially progress, right? This is forward progress of civilization, if you will. And I think the challenge is to sift through. So here's the challenge: data are cheap. You are getting, I'm while I'm talking to you, I have 14 feeds coming. I've got 14 different. You probably have more feeds coming in. To be able to sift through the data, to be able to connect those dots in a way that makes sense. I think that's where the challenge is. But yeah, I think we have benefited from this wealth of information data. Whether it's digital media, social media, whether it's health, electronic health records, bioinformatics. And I think if we can leverage that to good, I think we will be in good shape.
Yeah, I remember very clearly in the development of one particular diagnostic, coming into a room where the essentially the scientists were just saying, "Look, I mean, the data is so cloudy right now. We can't see anything. The picture that should be there isn't there. What we were expecting to happen isn't happening," and this, painted a very bleak future for what we were working on. And I remember the bioinformatics team saying, "Well, so you want rid of that?" Yes. And then they typed a couple keystrokes on their computer, and on the screen, the picture became a little clearer. And they said, "Yes, but there's still some of this and that." And they removed whatever that was, and all of a sudden the picture became a lot clearer. Now this major roadblock that we had, 30 seconds ago, with proper, data tools, we're again able to sort of be very selective about what we want to see. And I understand your point exactly, that it's great to have all of this data, but at times it's also, not necessarily enabling where we're trying to get to, and we have to have the tools and capabilities to sort of filter for that and find the information that we're actually looking for.
Absolutely. I think what you mentioned is something called machine learning. So there are algorithms, if you will, that can. So If you show me more than 15, 20 bits of data, I'm lost, right? I'm already, it's just too much stuff coming at me. But machine learning algorithms, they can take more data, the better, and they can start, hey, there's a pattern here, there's a pattern there, something's going on there. But where humans, where you and I still play a role is actually making, because machine, it doesn't know, everything's one or zero. It's a very binary type of environment that, to actually make sense. Okay. And then to put it into perspective, okay, this is liver disease. This pattern makes sense that it's not just some random something that somebody formed. So I think a collaboration, if you will, between machine learning, artificial intelligence, and humans, I think there is a lot of good to happen over there.
Absolutely. So, Prakash, let me ask you, what inspires you?
Possibilities. The possibilities today are not only endless, but I think they're within grasp. Okay, because like I said, technology is at your fingertips, and how you use them, I think you can make a lot of headway. Let me just give you an example. There is a show back in India, and it's almost global. It's called Indian Idol. There's American Idol and all that. Today a kid in some rural some place, no place, wherever, can literally, if that kid has talent, they can speak into their iPhone or whatever and upload it, and that talent can be recognized by other people. So I think the what inspires me is that the potential today is reachable. It's actually it's not some distant Wizard of Oz way out there. It's actually much closer, if you will. I think that really inspires me. Yeah.
What concerns you?
Concerns me is my own age. Okay, the runway is getting shorter for me. I wish I were 20 years. No, what concerns me is uh, misuse of everything we talked about. Getting like they say, the smartest people today are trying to get you to click on a button and buy something. So I think misuse of an opportunity, not putting it to good use. I think that may concern me. Everything else that's going on in the world concerns me, but we that's something we can't address right now. So
Yeah.
yeah,
Well, on the digital marketing side, we're very driven by data and the things that people do. And I get asked this question all the time. Look, I want more people to click this button, and the problem is that it's still a human experiment, and there's parts of things that you can clearly understand and see that drive human behavior. um, we certainly are more apt to solve problems than we are to buy features. And so those understanding those basics definitely gets more people to click the buttons, but you still have that question, will they? And there's a part of this that you still have to put it out there to know whether or not if they will or they won't. So yeah, I
absolutely. Yeah.
agree with you. Last question: what excites you?
Excites me is the next generation. I think what kids are doing today really excites me, and that's what I said. I wish I were, you know, 50 years younger, I guess, not 20. There's so growing up. I can't speak to you, but growing up, when you ask me what is Python, it's some snake. But if you ask a kid today, what's Python? No, it's a coding language, right? And I think that excites me that there is so much potential that the younger generation today is tapping into. Yeah, I forget who said Louis Armstrong, right? They'll know things that I will never know. I think that excites me, the potential that what the next generation can bring to the table. I think that really gets me going.
I do worry. I mean, on that side, and I've said this about AI, that I do worry about the the one feeling that I remember very clearly going through school are those moments when something clicked, when all of a sudden you started to understand why did I learn these five things before learning this new subject? Why is it important that this thing happen before the next thing? And I mean, as important as I think AI is, there's a part of your brain that has to be turned on, and it only is turned on through facing the challenge of having to intellectually think about something first and then work through it as a problem. And now all of a sudden, if you can accelerate that, will those learnings come faster, and will they be more profound because of the fact that the students of today have learning that they gain through artificial intelligence, or do they lose something in the end? And we won't know that until we get to that future point and look backwards, unfortunately. And so I think that just could be in really interesting experiments as well for the future.
Absolutely. I totally hear you. I think today some of the foundational underpinnings, they are definitely missing. Everything is just, you punch something in, here's the output. It's a black box. Nobody understands how it came to be. But the way I'm a hopeless optimistic. I don't know about you, Don, but growing up, I'm sure the generation before me or the one before that said, "Hey, this fellow, he's doing everything on a computer. He doesn't know how to sign his name or write in cursive." But that's what so yeah, today kids are or whoever they're using AI and all that. I think when you and I turn over many years from now, I think there'll be a new generation in place or generation alpha. I just learned yesterday that generation beta is already being born. I was just getting used to alpha. So I think it's just that's how it is, and I think we can fight it to some extent. We can question it. I hear you, but I think it's going to be a losing battle. It's I think digital media, AI, or whatever. I think 100 years from now, who knows? I don't think anybody will be doing math on or algebra on a piece of paper and pencil and all that. So it's what it is. So, yeah.
Yeah, yeah, for sure. I remember very clearly being forced to manually work through math problems before I could get a calculator.
Yeah,
I can
long
remember
division.
exactly.
Long
I can
division.
remember very clearly having to work through these things as well. And yeah, as scary as it might be, who knows? Maybe there isn't a circumstance where people, in the future, aren't as able to work through problems because of AI. I have no idea, but it'll be interesting to see for sure. Well, Prakash, thank you so much for being on the Life Science Success Podcast. I greatly appreciated you being here and talking about everything from small molecules to cell-based therapies. It was great to kick off the year, with an interview with you, and so I greatly appreciate it. Thanks a lot.
Thank you, Don. I really appreciate. I enjoyed dialogue and sharing whatever little wisdom or experience I have. I think if I had to leave the viewership with one thing, I'm just going to say that entrepreneurs and leaders they are goal driven. They don't let the small things bother them. They jump at an opportunity, they grasp it. They don't let sidebars, sidetracks. Boy, it's just part of the thing. So I think. If I had to just give one piece of advice from a small business or entrepreneur type of perspective, seek your goal, try very hard to achieve it, learn from your mistakes, and don't let the noise distract you. Happy New Year, and I think we're off to a good start.
Thanks a lot, and same to you.
Thank you.